presenting a little bit on some solutions, specifically one in particular that's related to my PhD of declining fish stocks. So I am a PhD researcher at UWA, but to start with, I'll just give you a little bit of background about my career path anyway. So I did my Bachelor of Science at Murdoch, and I did a double major in marine science and conservation, wildlife and biology. So one of the really big benefits about this type of industry is that you can do a lot of volunteer work so that you get a really good sense of the areas of the industry that you do or do not enjoy. So then you can, go into those areas and explore them a little bit better. So with some examples of that I did after my bachelor is I'm going to Exmouth, and in that bag there we have some coral, and we put sorry, there's a little bit of feedback. Hopefully you can hear me. So we take the coral and then we scrape off the algae there so that we can look at the algae abundance as a food source for fish. Then also in the Seychelles, this is their island archipelago off the coast of Africa. And here is a
Isobel,
picture.
sorry to butt in.
Yes.
Sorry. It's Cecile. Lauren, is your mic muted? It's really, there seems to be a problem.
Yeah, it must be us. I will do that now.
Thanks a lot. Sorry to butt in.
Okay, cool. No, that's perfect. It was a little bit distracting. So let me just bring it back up. Yeah, so in the Seychelles, we had a coral nursery there, and that's just us scraping over the macroalgae off that coral so that they continue growing so that we can transplant them. And then in Albany, we looked at plastic abundance found on beaches. So this is a really topical area of research at the moment. I also did turtle tagging at Barrow Island so that we could assess how light pollution is impacting the turtles laying their eggs. And I also did some volunteering at Rottnest Island looking at the intertidal rock platforms and the species diversity across there. And then saving the best till last, I also did some research in the Maldives. So these are manta rays, and it's actually a mating train. So we have the female at the front and then the following about 4 or 5 manta rays behind the males. And underneath you will see that there is some black dots. So they're the individual fingerprints basically for the mantas. So they allow us to identify each manta. So from this, I decided to actually go back to uni and do my Masters of Biological Science, and I specialized in marine biology. I won't really go into what my thesis was on because it's actually linked to my PhD, which I'll be talking about today. So I'm hoping to finish this by the end of the year, but again, my project was in the marine biology space. So I thought I'd start out by giving a little bit of a really big global background that isn't quite linked to fisheries to start with, but then we'll slowly get there, so just bear with me. So when we think about threats to the environment, we normally think of things such as energy production, transportation, deforestation, urbanization, and industry. However, one sector gets majorly overlooked. So our need for food poses one of the biggest dangers to the planet, and the environmental challenges posed by the agriculture sector are huge, and they're only going to become more pressing as we try to meet the growing need for food worldwide. So the current population sits at about 8 billion people, and food insecurity affects 820 million people. So these are people without access to sufficient, safe, and nutritious food. And this is the equivalent to the combined populations of Canada, the United States of America, and the European Union. So I think we can all agree that that's an alarmingly large number of people. But by the year 2050, the global human population is expected to exceed 9 billion people. So this rapid increase in human population places a great pressure on food sectors to maximize their production whilst also reducing waste so that we can conserve the world's remaining resources. So with this, we'll need 56% more food than what we are currently producing. And we also have an issue that 50% of total food weight is wasted that is currently produced. So to put that in example of Australians, it's the equivalent to every Australian 1 in 5 grocery bags that they purchase at the supermarket going directly into landfill. So how can we feed our growing population without impacting our world's remaining resources? So to do this, we will need to increase food production, but we will have to do that without expanding our agricultural land. So already 50% of vegetated land is used for agriculture. And this is not just limited to crop farming, but also all agricultural practices. Also, in a new study, it was shown that food systems generate about 85% of total global man-made greenhouse gas emissions. So if we were to break down that 35% share, production of animal-based foods, so that's things like meat, poultry, and dairy products, but it also includes things like growing crops to feed the livestock, as well as pastures for grazing. It contributes to 57% of the emissions linked to the food system. So we've exhausted our land resources essentially, and we can't create more emissions. So what are our other solutions? So is fisheries potentially a solution to this? So fisheries sector plays a crucial role in providing food, income, employment, an array of social and cultural values to many. So in most instances, fish provides an affordable, nutrient-dense food source, and it covers minimal intake requirements and enables access to nutrients that are not readily available in other animal protein sectors. So an example of those nutrients is unique long-chain polyunsaturated fatty acids. So in other words, omega-3s and 6s, which I'm sure we've all heard of before. But the presence of these macro and micronutrients that we find in fish can also facilitate health benefits in both child development and adult health. So it's really important that we do have access to them. Examples of things are as preventatives to an array of cardiovascular diseases. So for example, coronary heart disease. But most fishery stocks are assessed as overall maximally fished, as you've apparently learned about already. So this is a graph from the Food and Agricultural Organization of the United Nations that we refer to as the FAO, because I've got a few more graphs going forward from them. So this figure here shows that the underfished proportion, let me see if I can get the laser up. So the underfished proportion of world fish stocks, the maximally sustainably fished, as well as our overfished. And as you can see, over time, our percentage of underfish has decreased. The amount of the sustainably fished has stayed roughly the same, but the amount of overfished has definitely grown a lot. So this is pretty alarming because it results in limited marine sources and further pushes us towards a future of potentially reduced food insecurity. And also declines in fish stocks are estimated to leave about 845 million people with these macro and micronutrient deficiencies that I just mentioned, and especially in low latitude developing countries. So if we go back to my earlier slide where I showed the equivalent of the combined populations of the United Nations, the European Union, the Canada, and the United States of America, it's about the same. So that's really quite concerning. So to add to this now, we also have that 33% of fish stocks have fished at unsustainable levels. But there is there's different types of fisheries. And so this is where I will start touching on aquaculture. So first we have capture fisheries, which is mostly what I just mentioned, where we capture wild fish from the ocean. But aquaculture differs as we first grow and then rear the fish, and then we capture them. So aquaculture is the most rapidly growing food production sector globally, and it's increasing by about 8.5% per annum. So it's offering a solution to our diminishing fish stocks. So again, this is a figure produced by the FAO, and we have got the capture fisheries in blue here and the aquaculture in pink. And so from about maybe 1990, you can see that capture fisheries starts plateauing with a few peaks and troughs there, but aquaculture on the other hand is just increasing. So this is really great. And we can also see that there's a few different types of aquaculture we can get as well. So we have got finfish, which is in the blues. Then we have crustaceans, mollusks, and then other aquatic animals, but also algae is also beginning to take off. But then again, we do have a small issue within the aquaculture sector that we do obviously need to feed the fish if we're rearing them. And currently, many aquaculture species are carnivorous, so they have high dietary protein requirements that are often delivered in the form of wild caught fish, which we refer to as fish meal. So a ratio that was posted by the FAO predicts that about 5 units of wild biomass are required to produce 1 unit of farmed aquaculture biomass. So this is quite concerning. And if we were to put this into a little schematic, we have our fishery resources in decline, as we just mentioned. And then aquaculture is showing us that we could potentially get a solution out of that because we're farming fish instead of removing them out of the ocean. But then those fish have the high dietary protein requirements, so we're in this negative feedback loop. And in order to break away from it, we can find alternative protein sources to fish meal, because then this still means that aquaculture can help solve our problems of diminishing food worldwide, as well as reducing our reliance on those wild caught fish as well. And the reason why we would focus on aquaculture and refining that industry is because of a thing called food conversion ratio. So that's the biomass of food taken in by an animal to produce the edible biomass out. So for example, fish require about 1.1 kilos of feed to produce 1 kilo of fish that we can eat. And if we were to compare this to the beef industry, 10 kilos of feed are required to produce 1 kilo of beef. So aquaculture is definitely a more sustainable option in comparison to our land-based alternatives. However, that doesn't mean that there isn't refinements that still need to be made. So this was what a fish food normally looks like. And if we were to break that down, there's several components to it. So we have got our plant dry matter here, and that's things like wheat, and then land-based animal ingredients, so that's things like poultry. And then we have marine animal ingredients, and this is our fish meal component. So these percentages here are an example of a barramundi diet. So fish meal takes up about 15%. We also have marine oil, so that's fish oil, land-based animal oils, so chicken oil, and then other micro and macronutrients that we add to make them nutritionally balanced. So previous research has looked at alternative protein sources, and it's largely been focused on plant-based matter as a substitute, and particularly things like lupin, soybean, and rapeseed. But there are concerns over the suitability of plant meals, as they tend to have anti-nutritional factors and the potential to cause inflammation of the digestive tract and a decrease of meal palatability. So decreased palatability means that it doesn't taste too good, so the fish doesn't want to consume too much of it. And then if we have issues like inflammation and anti-nutritional factors, it's very similar to what happens for a human. So if you have inflammation, it's harder for you to eat and digest, and you won't be taking up that nutrients. And if it's not nutritional, it's the same as you basically eating Macca's every day. So research has begun looking at other alternatives such as bacteria, algae, and insects. And this is where my PhD comes in. So my research is focused at looking at insects as an alternative protein source in aquaculture diets. And I look at an insect in particular called the black soldier fly. So as I mentioned earlier, we do have a few issues associated with our food production systems. And there's an array of benefits that come from the commercial production of black soldier flies, and one of which is the facilitation of a circular economy. So for those of you who might not be aware, a circular economy is a model of consumption and production, and it's based on 3 principles. So we design out waste and pollution, we keep products and materials in use, and we regenerate our natural systems. So basically, we're ticking off the 3 issues that I presented earlier. So to talk you through that briefly, we have an input of an organic waste. So that's things like the fruit and veg that are considered too unaesthetically pleasing for supermarket sale. But it can also be things like manure. And then that's collected and transported to the insect farm, where the insects feed on it, and they bioconvert the waste into their own biomass. So this is great because studies have shown in the case if they were fed manure, it reduces emissions of the ag sector, with manure mass reducing by up to 50%. We then can produce products from this, and the idea is to reuse them back into the agri and aquaculture sectors to complete that circular economy. And by doing this, we essentially want to produce sustainable agri products whilst reducing waste between paddock to plate. So the three products that we can get from here are frass, oil, and protein, and this is what they look like. So first, frass is the excess, the uneaten food, the larvae's feces, as well as the exoskeletons of the dead bugs or when they have shed. So that's typically used as a fertilizer at the moment. Then we also have the oil proponent, so that's when we can extract oil out of the larvae. And this is used in industries such as biofuels, potentially. Obviously, a lot of research needs to go into that, but at the moment it's showing to be quite promising, as well as even just on smaller scale things like soap. And then we have got our protein proportion. So this is what we're most interested in, as the protein for the larvae is actually really comparable to the protein that we'll find in fish meal. So we've got the same protein level and fat level. So this is our first indication that potentially we can use this insect protein as an alternative in aquaculture diets. So how do we actually test this? So I'm just going to talk you through what my PhD overall looks like. So we have our project development, which is coming up with our trials, and then we have our data collection. So we feed the larvae to our fish, and then we sample them over a period of time, so we can get a nice growth curve. And then at the end of the trial, we take samples, and from them we analyze them, both doing nutritional analysis as well as chemical analysis, but also we have to look at the computer analysis to find if there's any differences. And then we go into our thesis write-up, but I'll explain that a little bit more in depth in a moment. So my PhD is looking at not only using the larvae, but as well as seeing if we can use the frass. So I'm feeding the larvae as a protein source in barramundi and marine diet so that we can look at finfish and crustaceans. But we also had the idea of feeding frass as an alternative protein source because, as I mentioned in the earlier slides, there's many components to fish meal. And we do have those plant-based components that, for example, are wheat. So we can make the same argument that we do with fish meal is why would we grow wheat to feed to fish to feed to humans when that can go straight to humans, especially when we are predicted to not meet our wheat supply. Our demands in the growing future as well. So at the end of our trials run for about 3 to 4 months at the moment. And so at the end, we analyze a few variables. So we take the weight and length throughout the trials. So essentially, basically, we put fish into a tank. We have multiple tanks, and we have multiple tanks allocated to a treatment, which is the experimental diet. And each experimental diet has a different proportion of insect added into it. So we're wanting to see how much black soldier fly we can add and whether we can add it at all. And so at the beginning, we take the weight. So we have with a nice starting weight and all the fish weigh the exact same. And then over time, we take those measurements so that we can see if there's diets that are underperforming or performing better than our control diet, which is a commercial diet that contains no larvae. So we do this so we can see a growth rate, and we pit tag our fish, which means that they have a little microchip in them that you can scan so that you can tell individual fish. And that allows us to not only see the tank as a replicate, but also the fish. Then we take some biochemical samples at the end. So we do the viscera, which is basically the intestinal, everything that is within the fish, basically within their stomachs. And then we can get a viscerosomatic index from that, which shows us how much fat deposition there is. This is important because when we're feeding these fish a diet, we would like that energy that comes from the protein source to go into growth instead of into fat. If it's going into fat, it shows that there is potentially anti-nutritional properties and they're not being digested and absorbed. From the viscera, we remove the liver and we get the hepatosomatic index, and the liver shows us an energy store. So that shows us something quite similar, and those two indexes complement each other. We also take a histology sample. So that's when we take a little tissue sample and we fix it onto a slide and look at it under a microscope. I'm not sure how much you guys have done potentially in biology, but it's so you can look at the cell structure of these organs. Then we also remove the intestine and we take a microbiome sample. So that's so we can look at the gut flora and fauna. So we can see if there's good if there's good bacteria, good fungi, good viruses hanging around, as well as the same histology sample. And then lastly, we do a blood test. So this allows us to see the overall health of the fish, much similar to what you would get as a human blood test as well. Then we also do a health challenge. So this is when we inject the fish with a disease that is prevalent to it. So I work with barramundi, and the disease is Streptococcus ineae, and it's really prevalent in aquaculture settings for barramundi. And the reason we do this is that we would like to see whether black soldier fly either causes it boosts the immune system and the immune response of the fish, or whether it hinders it or just stays the same. And then lastly, we remove the fillets from the fish and we weigh them. So we get a fillet weight from this, which is the sellable product. And then we also do approximate analysis. So this allows us to look at the nutritional profiles of the fillets. Approximate analysis basically tells us things like how much fat, protein, and carbohydrates are in the sample. And the reason we do this, and I would argue that the fillet analysis is probably one of the most important, is because we can grow a fish that's double the size, but if it doesn't have any nutritional value, or if it doesn't look too good, then we basically aren't going to be able to sell it to the consumers. So that nutritional profile allows consumers to ultimately make decisions on the products they do or do not purchase. So all of these different components are basically a puzzle piece, and if we don't consider and weigh all the evidence derived from the multiple biological endpoints, then it can lead to the reporting of false positive or false negative results. So the best way to kind of think of it is we have to consider our growth, our biochemical health, our gut health, our immune response, and the consumer perception of these end products to get a better idea of the degree of suitability of black soldier fly as an alternative protein source. So this is just a little snapshot of a routine that we have, a standard routine during trial. So we have feeding here where you can see our individual tanks. So this is just a little snippet basically of the systems we have. So we have got 24 tanks divided between these two systems here. Each tank has water inflow through these little tubes here, these aeration pipes that are lifted up because these are really important for fish health. And then we also have the fish within the tanks, obviously. So during our sampling, during our trials, we go around to each tank and feed them their experimental diet each day, and then basically repeat every day until the trial's over. I didn't touch on this today, but this is just only one example of the different aquaculture we can set up. So this is a recirculating aquaculture system, also known as a RAS system, and this is an on-land system. So it means that the water that we're basically recycling the same water. So this can have benefits compared to a flow-through system that we have on land as well, where you pump water from the ocean and then dump it into a water treatment. Whereas this allows us to keep going, so it's less water use. But there's also another version, which is sea pens or sea cages, which you can have in open bodies of water in the wild, where obviously the water flow is just natural. But again, there's pros and cons of each system, which potentially you'll probably touch on through when you cover aquaculture. But then we also have our sampling. So this here is a measuring board with a fish on top, as well as some scales. So what we do is we pop the fish down, we scan it for its pit tag so we can get the right ID for the fish. And then we take its length and its weight, and then we use that data later on for those examples that I gave earlier. And then this is just a bird's eye picture of that with the barramundi. So we will go take the measurement here for fork length and its weight that will be off the screen. And then we just do daily monitoring. So these are some juvenile marine that we used in our trial. This is on a day where we empty the tanks to clean them because they should be white, not green with algae. But we remove them out of the tank and we're able to, we coincide it with our sampling, so it's less stress for the fish. but it allows us to see if there's any health impacts as well. But we also just do daily monitoring as well to make sure that the water temperature is correct, there's the right aeration, and that nothing looks amiss that could potentially stress the animals or impair their health. So this is just an example of the data analysis. This is the size of the barramundi that I started my trial on, which is 35 grams. And then after 13 weeks, I ended at 370 grams. So this is a fish that has been fed that black soldier fly diet. And these were really great results because it's basically suggesting that black soldier fly is a suitable alternative. But as I mentioned, we do have those extra pieces to the puzzle that we need to consider. So what I didn't mention earlier is that I look at a few different ratios and experimental diets using black soldier fly. And the way that we did this is we wanted to answer 3 main questions. So the first one that I'll mostly touch on in this example is our optimum inclusion level of black soldier fly. So we included black soldier fly in a stepwise inclusion that went from 5, 10, 20, and 40% of the total diet. So as I mentioned before, fish meal takes up about 15%. So those inclusions of 40% is quite a lot of the diet. But we wanted to basically see how much of this product we can actually use. So we already know that growth was successful, but also when we looked at blood, we found that everything was within healthy parameters, and that histology was within healthy parameters. So this is really great because blood and histology complement each other incredibly well. So if we don't see any differences in these 2 areas, it means that all fish were considered to be healthy. Then at the bacterial trial, we found that the highest inclusion level reduced survival. So this is suggesting that that 40% inclusion level was just a bit too much black soldier fly. And then lastly, when we looked at the consumer perception, we found that the fillets were highest, for the highest inclusion, had an increase in bad fats and a decrease in good fats. So this is something that a consumer wouldn't be wanting to look for. But to go back to our circular economy, what my PhD hopes to look at is that inclusion of, in this case, for the results I just presented, black soldier fly protein in use in aquaculture diets to play their part in the completion of this circular economy. And from my research, we have found that black soldier fly is a suitable protein source. However, there is the caveat of the highest black soldier fly inclusion, showing early signs of impaired health and nutrient availability. But as I mentioned before, the percentage of fish meal that we have is 15%. So our second inclusion level, just the one down for the 40% was 20%, and we found that there was no impairments to health and nutrient availability when we had that. So that's a really promising results and really exciting for this industry moving forward to see if we can keep refining that and find suitable protein sources. So to go back to our main question of where will we find enough food for 9 billion people without using more land resources, using more ocean resources, and creating more emissions, we think that black soldier fly as an alternative protein source in aquaculture can really help us on our way to doing this. And that's all I've got for you today. Thank you very much, and please let me know if you've got any questions.
Awesome. Thank you so much, Isobel. That was really good. Everyone,
That's okay.
How much longer have you got left of your PhD?
Oh, hopefully no longer than December.
Yeah, okay. Yeah, good luck. It was really interesting. Thank you. I've got a couple of kids with a couple of questions. Rubes, do you want to go first? Thanks.
I'll just stand in front of the camera.
Yeah, just have
As if she's a person.
Hello.
Hi. What are some of the best ways to seek out volunteer opportunities?
Definitely by just having a look around for some companies, or also probably the best way. Are you talking like aquaculture in general or just marine science?
Just marine science, I guess.
Yeah, so one of the best probably ways for volunteers is to go through universities, and you can look for the marine science academics, which will always be listed on the contact pages of universities. And basically they all have their own unique research areas. So if you find someone that has got something that an area that you enjoy, if you send them an email and see if they have research like volunteer opportunities, or if any of their students, so a lot of them will have masters and PhD students that will definitely need some help depending on their project. So that's probably the easiest way while you're still in school. But once you're at university, there's Facebook groups you can join where people post where they need volunteers, and there's also some programs that you can pay to be a volunteer on, which is up to you whether that's something that you want to do. But yeah, you can get some cool experiences either way.
Thank you. And then also, what has been your favorite experience working in the marine field?
I think it's the diversity of projects and the applicability of the skills you learn and the transferability. So I have gone into the aquaculture area, which was something that I didn't really ever see for myself because I didn't really know anything about aquaculture before I started my masters. But when I started my project, I was kind of torn at the time, being like, do I want to do an aquaculture project? Because what about it? That's not the area I want to stay in. But you learn so many skills. So for example, in mine, you learn how to run a project, you know how to do all these different types of analysis that can be transferred into other aquaculture areas. So for example, the microbiome, people are looking at that for whale sharks at the moment. So yeah, you learn a skill and you can transfer that into completely different opportunities, which I think is quite cool.
Can I just butt in? Can you mute again at the school? Because there's lots of feedback. Thanks a lot.
Yeah, no worries. Obviously we need to be unmuted when Luca will ask another question, and then I'll mute it now once he's done it.
Thanks so much. It's just in the reply. Yeah.
Yeah.
Hey, how did you decide to choose a black soldier fly rather than other insects?
Yeah, cool. Well, so for my case, it was already written into my project grant. So for full transparency, I personally didn't get a choice in it. But the reason why the grant did choose black soldier fly is because of that circular economy that I mentioned before. So in other insects, there are benefits to it. For example, some insects are able to convert plastic into a more digestible breakdown. But for black soldier flies, they only feed as a larvae, so you compare that to a house fly. They feed as adults as well, which makes them a nuisance fly. It also means that they are considered a pest and a disease vector. So that's not the case for black soldier flies. So if we were to have a breakout of them, it wouldn't really have much of an impact to our vegetation and our flora and fauna. Black soldier fly are also considered naturalized to every continent except for Antarctica. So that's great because it means that our research is able to be easily transferred into other countries as well. But mostly it's that circular economy, and they're able to reduce waste into its own protein. And then we are able to alleviate greenhouse gases and all the waste, that's excess waste that's been generated.
Cool, thank you.
Thank you.
And just one more question, Isobel. Thank you.
Go.
Hello.
I just wanted to ask how you go balancing your life with everything else, as well as doing your PhD.
Oh, thank you. Good question. That is a tricky question. So with aquaculture, well, for my project, I actually did, I counted it, over 560 days of field work in a row. That's only just because of the nature of my project specifically. If anyone wanted to pursue aquaculture, it wouldn't always be like that. But one thing you really had to learn was balance. So it's really important, I think, that if you are considering any type of research, as well as, I guess, any job, is to really make sure that you give yourself time for a rest period, because We notice a lot, and we have a lot of discussions about this in our PhD cohort, is that if you just go hard with your research and with your field work, because in environmental sciences, a lot of the time you are out there collecting stuff and doing quite long hours and a lot of hard work. If you don't balance that, then over time, the hours that you put in kind of become redundant because you're not working at your full capacity. You're not able to really you get brain fog, you get burnout. So by giving yourself those timeouts, it means that you work better over time. So yeah, it's just important to remember that, and I guess not feel guilty if you are wanting to have a break and go do something fun. So yeah, I think it's just reminding yourself that it works better in the long run.